Greetings to all of the audiences. My name is Reina, and today I will present you my research plan about virtual screening of Plantensimycin-based inhibitor targeting for beta-ketoacyl acyl carrier protein synthase for tuberculosis therapy. And my research will be supervised by Professor Dr. Usman Sumafran Tambunan. So today I will show you the introduction until the timeline of my research. And let's begin by seeing this surprising fact. In 2020, 1.3 million people died by tuberculosis, and this is not the only concerning number according to this disease. In fact, in the same year, 5.8 million new cases were found, with Southeast Asia as the region with the highest case, which is 43% of all of the cases around the world. Meanwhile, our country, Indonesia, placed in the third as the highest new cases for the statistics. As you can see in the picture on the right, the 8 highest countries, and India is in the first rank. So as you can see from the statistics, I believe that this disease is urgent and we should find the solution of this disease. So one of it will be finding the drugs. We should also see the drug development before. So tuberculosis. This disease is caused by infection by Mycobacterium tuberculosis using air transmission. And currently, there is a vaccine that has been used for around 100 years called Basil Calmette-Guérin, and the first-line drugs for this disease are isoniazid, rifampicin, ethambutol, and pyrazinamide. But due to the mutation and the resistance of these bacteria, the evolving bacteria had caused the resistance, such as rifampicin resistant, multi-drug resistant, and extensively drug-resistant tuberculosis. So after knowing the urgency of this disease, we should move on to the discovery of the drug. To find the suitable or the best option for drug or for finding the drug, we should see what important component of this bacteria, the mycolic acid, one of the components in Mycobacterium tuberculosis that has role in protecting the bacteria. This component is biosynthesized by KSA enzyme that elongates the fatty acid. And after knowing the target protein, we should also know the potential ligand. I propose to use plantensimycin as a potential antibiotic since it has inhibition activity against fatty acid synthesis enzymes. And to make my research be purposeful, we will move on to what we should identify in this research. First, is there any potential plantensimycin derivative as beta-ketoacyl acyl carrier protein synthase enzyme inhibitor in Mycobacterium tuberculosis? Second, how is the interaction produced between the inhibitor and the enzyme? And the third one will be, how are the pharmacokinetics and toxicity process of the derivatives? And also, we aim to define the structure of the plantensimycin derivative that act as the enzyme inhibitor, show the interaction between the enzyme and the inhibitor, and describe the pharmacokinetics process. And also, I had hypothesized that plantensimycin derivative can act as antibacterial agent towards the enzyme. This derivative is also able to interact with amino acid residues, and the derivatives are also suitable for drugs since it has low toxicity and low side effect. So after understanding the base, we also need to know more about this disease, about the compounds, the principle, and also the method. And we move on to literature review. So this research was based on the first one will be in 2006. Plantensimycin has antibacterial activity against gram-positive bacteria, and followed in 2009, it had been proven that this derivative has antibacterial activity against Mycobacterium smegmatis and Mycobacterium tuberculosis. And this also supported by 2011, Plantensimycin and its derivative, Plantensin, has antibiotic activity against Mycobacterium tuberculosis. And for the enzyme, the inhibition had been proven can be done by thylatamicin, one of the first-line drugs compounds. And for the computational screening, it had been done to see the inhibition process or the interaction between small molecules and the enzyme. And after knowing the base of our research, let's move on or let's understand more about the disease tuberculosis. So this disease was first found by Dr. Robert Koch in 1882, and the cases of tuberculosis was rising due to the acquired immune deficiency syndrome or AIDS and the environmental problem in developing countries in 1980. The conditions of this disease are pulmonary, extrapulmonary, and latent tuberculosis. And this disease has four stages of infection, starting from lymphatic circulation or the bacteria were spread through lymphatic from the third week after the implantation in the lung, and the highest stage will be extrapulmonary lesion, or the bacteria attacks other organ than lung in and it will be done for 3 years. And after understanding the disease in general, let's move on to the Mycobacterium tuberculosis itself. So this bacteria is a soil bacteria and gram-positive, can be found in several species. And the most important component will be the cell wall that are coated by around 200 cans with mycolic acid as the component with permeability and virulence support. As you can see in the right, the mycolic acid is the outer cell wall of the Mycobacterium tuberculosis. And also this component, the mycolic acid, is biosynthesized by one of the enzymes, which is KSA or beta-ketoacyl acyl carrier protein synthase. This enzyme had been evolved due to the mutations and have several mutagenesis in some positions of codon, including the cause of increasing resistance of isoniazid. As you can see here in position 66, 269, and the next will be in 212 and 413. And after knowing one of the resistance towards isoniazid, let's move on to the specific or what codon that we use or what mutation that we use. We will use the mutation in codon 312 that causes resistance toward isoniazids since it has the highest isolate percentage in the research concluded here. And also, after understanding what enzyme that we should inhibit, let's move on to the next one that will be the natural product that we use. So this is the structure of Plantensimycin derivative that can be found in uh, Plantensis, and this natural product is selective towards the fatty acid um, synthesized enzyme. And for the principle of drug discovery will be enzyme substrate active site traits, especially competitive inhibition in that is beneficial to a drug discovery. And the method that will be used will be in silico method or computational simulation. And after understanding the basis, we'll move on to methodology. This research will be done in February until June 2022 in Department of Chemistry, Universitas Indonesia, and the tools will be used to obtain the 3D structures and also to analyze and prepare the physical as well as chemical traits of the ligand and to analyze the ADMET. And the materials will be got from branded and zinc database. And here are the research tips that will be started from enzyme preparation by downloading and doing protein optimization and ended by process efficiency and continued by ligand preparation from downloading and screening using Osiris Data Warrior and MOI and preparing it using MOI. And the third step would be docking using MOI also, and the final step and one of the preparation and also the research itself, and will be ended by the thesis defense. So thank you for your attention today.